Well, hello, this is Miss Dagenford, and we're going to be looking at the membrane structure and function. And the membrane structure and function is something that you have heard before in honors biology or regular biology. And if you took anatomy last year, we went over it as well. So hopefully none of these are completely new. It might be a couple of years since you've had it, but there should be some memory of these. So by the end of this video, you should have a good understanding of the phospholipid molecule. You should be able to identify the major parts. You should be able to explain how they orient themselves with each other and explain why. You should explain how the membrane behaves with regard to temperature, composition, and how the two relate to each other. The fluidity of the membrane, the membrane proteins, and we're going to be able to apply this to what we've already learned about the proteins as well. So let's get started. So if you were to focus on this little picture right there, you might recognize our little phospholipid. So this picture is looking at a phospholipid. And again, hopefully you were thinking, oh, yes, I remember those. And what's interesting, there are going to be some things that you should recognize. For instance, this long molecule is part of a fatty acid. So these are fatty acid tails. And then this is a polar, we call it the polar head. Now, what that means is it is the one that mixes with the water on the outside. And then down here, we also have another fatty acid chain. Now, we should notice that one has that very distinctive bend to it. This means that it is unsaturated. And then over here, or I could point this one, this is saturated. Now, it's going to play a role in how the membrane moves, its fluidity. You can see over here, these phospholipids stack very nicely next to each other. Now, the more they stack, the less fluid movement they have, the less fluid it is. You can almost think of like a gel setting up. If they're closer together, they're packed tightly, and that limits their movement. And then what we're going to be looking at as well, the proteins involved. And so very commonly you'll see these like globular purple structures, and they're going to be proteins. Now you want to remember that the inside of the membrane, where the tails are, these are nonpolar. So let me go back and just add that. So these are nonpolar. Now they also have other words to describe this. So we have the polar head, that is called hydrophilic. And then the nonpolar tails are called hydrophobic. Now, the term is implying water. Hydrophilic is loving. So that's where they're getting that idea from, the polar head. So this is a polar group. It mixes with water well. The nonpolar tails, they will not mix with water. They are hydrophobic. Now, one quick word, phobic refers to as fearing, but I really want you to remember that the nonpolar tails, they're neutral. So they don't really repel the water. Water just doesn't mix with it. And so there's a separation. So there's kind of our brief overview. Polar head, and we can kind of divide it like right here. Polar head, hydrophilic, loving, water loving, and the nonpolar tails. The other term for it would be hydrophobic. And you can see the different fatty acid groups. Now, you might recall over here on the left is a beaker, and imagine I were to drop these phospholipids into a beaker of water. And if I do that, and then over here I have an after picture, what we would see is that these phospholipids are arranging themselves and of course arranging themselves. They're not doing this deliberately. This is just how the chemistry um, dictates their movement. The nonpolar tails are going to be excluded from the water, and then the tails are going to orient themselves in. Um, and then if we have a bilayer where we have water inside the cell and then water on the outside, that's what gives it its very classic lipid bilayer. Now, a bit about the history. Let me get rid of that. A bit about the history. They understood since about the 1900s. They had an idea that it was phospholipids. And then pretty soon they had an idea that it was two layers. They did some really interesting experiments on red blood cells. And red blood cells are really just a membrane and then nothing else inside. So it was pretty easy to use red blood cells as a mimic or as a model to understand the membrane. And so they understood that it was fluid, they understood that it was lipids, they understood that it was a bilayer. But they didn't understand how the proteins were involved. And so we have a quick word, and I still like using, even though they would probably not ask you about the Davison-Donnelly model, it's probably good to understand why they developed this process or developed this model. So Davison and Donnelly came up with this model, and it was colloquially called the sandwich model. And the sandwich model kind of says that the nonpolar tails are facing in, no problem there. And then they had to come up with, well, what's happening with the proteins? Where are the proteins found? And their model had the proteins on the outside, much like the bread of a sandwich. And so that was their model. Now, just to be clear, this is the wrong model. This is not right. But that's the purpose of the scientific process. They went through and they critiqued and they understood. And then eventually when Singer and Nicholson, these two scientists came along, they proposed what we now know or accept as the fluid mosaic model. Now, the reason that the Davison-Donnelly model was unsatisfactory is that proteins are very complex molecules. And in some proteins, you might have a region that is nonpolar. Let me redraw that. You might have a region of that protein. Maybe this region is nonpolar, but then ends up looking like a tree. And then this region up here is polar. And if you ponder it, this is really going to be unsatisfactory because the entire outside of the membrane would be bathed in water. In fact, you have the polar heads. So to then say that, okay, this region is polar and this region is polar and this region is polar, that's forcing nonpolar elements to mix with polar elements. And that's just not going to work. So Singer and Nicholson, one of the pieces of evidence was this really cool technique called freeze fracture. And the freeze fracture is where you dip it in liquid nitrogen, the cells flash freeze, and then you either drop the specimen or crack it with a knife, and you're hoping that you hit, notice the knife here, you're hoping you hit this sweet spot where the ice cracks, and as it lifts up, you're lifting up the two layers. And then notice you're going to get bits of the proteins that were embedded in the nonpolar tails. And that allowed them to see a very interesting phenomena. And then they used an electron microscope. And what they were realizing is they were seeing, see the little bumps here? Those are actually the proteins. And that led them to this nice aha moment of, got it, the proteins are embedded within the phospholipid membrane. And then that would give us a very satisfactory chemistry going on with this. So if we went back here, you would see that the outside of the protein here could be nonpolar, or maybe the whole middle section is nonpolar, but you have a way now to explain why proteins had nonpolar regions. Some, remember, nonpolar mixes with nonpolar, polar mixes with polar. So we can't have polar and nonpolar together. So here we have, this is the fluid mosaic model. This is kind of what we understand today. And we have our proteins, we have our phospholipid molecules, we have our cholesterol, and cholesterol does an interesting thing. It stabilizes the cell membrane. It can kind of work to keep the membrane fluid, but then also not allow it to get too fluid as well. So it's kind of this nice peacemaker, if you will. It allows some stability within the phospholipid membrane. Then we have here we have a glycoprotein. A glycolipid. You can see the carbohydrate groups, the green, and then that's attached to the nonpolar tails. You can also have a glycoprotein, and you can see the carbohydrate groups attached to the protein. The Ruffy R and Golgi would be involved in that. We also have these peripheral proteins that are inside versus an integral. So here's an integral protein, think integrated, versus peripheral. So this little guy would be a peripheral protein. And then on the outside, we have these proteins and carbohydrates. You want to think of them as a bit like glue. This is going to all stick together. And especially with the water and the other proteins, it's going to be all this nice stickiness that cements cells together. And then on the inside, we have cytoskeletal proteins, and that helps anchor and create the shape of the cell. So how did they figure out the fluid movement? Well, this was an interesting case. They took a human and a mouse cell. Now this, it was just, these are cells. They're not embryos. They're not babies. Don't worry. It was just two cells. And the important thing is they labeled with antibodies, and the antibodies were specific to those proteins. So these little pink and the little blue, those are proteins. But they were specific to mouse and human. And then we used antibodies that would attach to them specifically. So what you again get is this targeting, and we could then follow. And what they realized when they fused them together, they were initially separate from each other. So there was initial separation. The pink and the blue were distinct. But then after a few hours, what they noticed is that the proteins mixed. Now, what does this show? What this indicated is that the membrane is fluid, that the proteins can move within the phospholipid bilayer. And so you want to think ultimately that these membranes, you have this kind of this oil mix. And so these guys can scooch this way, they can scooch this way, they can go sideways very easily. They don't really flip-flop well. They don't flip-flop from inside to outside well. Now I want you to ponder, I'll leave you with this picture. Notice the one on the left, we have lots and lots and lots of unsaturated fatty acid. And notice how they're like bumping into each other more. And then here we have primarily all saturated. So this one is saturated, this one's unsaturated. And then down here we have our cholesterol involved. What we want to look at tomorrow is how the membrane is involved in kind of controlling its movement, controlling its fluidity. And then we're going to focus on proteins next. So have a lovely evening.